In this video, I want to talk about why mutations in DNA can cause such big problems. Let's start with a quick review. Remember, the whole point of DNA is to provide the directions to end up with proteins because individual organisms, you and many others here in the world, are made up of proteins both structurally and also the enzymes that help things happen. In order for the DNA, the information in the DNA or the information in the genes, which is found on one side of your DNA, and of course your individual DNA has many, many genes on it. But in order for those genes that directions to be turned into protein, we have to use mRNA and go through processes known as transcription and translation. This picture provides the overview of this process, which is known as protein synthesis, where transcription in the nucleus takes the gene in your DNA and makes a copy onto a molecule of mRNA, which then moves out into the cytoplasm and hooks up with a ribosome to undergo the process of translation, where the individual amino acids that are going to make up your protein are connected in the right order because of the order of bases on that mRNA. And the body reads those bases in groups of 3, known as codons, which then are matched up with the correct anticodon on transfer RNA molecules that are bringing those amino acids, in this case they look like little green balls, together where they are hooked together, eventually forming a protein. Remember, amino acids are the monomers of proteins. So then, what are mutations? Well, mutations are a change in the genetic code in the DNA. So genetic code is just another way of saying DNA. And it can be caused by a change in the sequence of the nitrogenous bases, that A, T, C, or G, that are found in the gene, because it's the sequence that actually determines the code of the gene. Or a mutation can be caused by a change in the chromosomes, by gaining or losing some part of a chromosome. If this mutation in the DNA occurs in gametes, which are the sex cells or the eggs and sperm in humans, then all the body cells of the offspring will be affected because, remember, organisms that undergo sexual reproduction start off as just one single cell, and that cell divides and divides and divides by mitosis to create the larger organism. So if something is mutated in the very first cell, all the cells will be affected. If later on in life a mutation occurs in an individual body cells, then only those cells will be affected. And that's where you get things like cancerous tumors that come up because of a mutation in a body cell, say a skin cell, that will develop into skin cancer. So where do mutations come from? Well, mutations certainly can be inherited. We all are walking around with mutations, and most of them don't really matter. They pass from our parents down to us because they are present in the gametes. But mutations can be acquired, and these are the ones we tend to worry about. There are various things in the environment that can damage our DNA. Some of the ones that may be familiar to you: smoking damages DNA, which then leads to a greater risk of cancer. Ultraviolet radiation in sunlight can damage DNA, again going back to skin and skin cancer. And then there are chemicals that are known as carcinogens because they promote mutations that can lead to cancer that are out there in the environment and affecting us. We also have mutations that just end up being mistakes that have happened as the DNA is copied. There's many protective devices inside cells to prevent these mistakes, but they do creep in, especially as organisms get older. So I want to go through some of the specific examples of mutations and give you some real-life examples of where these occur. So a mutation that happens in the gene that affects those nucleotide bases, the A's, T's, C's, and G's, are known as point mutations. And it's a change in one or more individual nucleotides. So that's going to affect the amino acids, and you're going to end up with a wrong amino acid being put into the protein. You're changing one thing, so it's called a point mutation. There are 3 different ways that these point mutations can occur. A substitution mutation occurs when a nucleotide is replaced. So instead of an A C is put into the DNA when DNA replication occurs. Or there may be an insertion where a nucleotide is added, actually making the DNA a little bit longer. or that gene of the DNA a little bit longer. And then there could be a deletion point mutation where a nucleotide is removed, making that gene a little bit shorter. All of these will have different effects on the end result of that protein that is supposed to be produced by that gene. A silent mutation is a substitution mutation which does not make a significant change to the final protein. Many of our amino acids actually have more than one codon or more than one code to cause it to be formed in the protein. So if a substitution mutation ends up with the same amino acid being coded for, it really is not going to affect anything, and that's why it's called a silent mutation. So for an example, on the DNA level over here in this diagram, if the original DNA is supposed to be TTC and it gets altered by a point mutation to be TTT, the ending mRNA codon, AAG, and AAA both code for the same amino acid. They both code for lysine. So either one of those codes will work. Even though there was a mutation, it doesn't have an effect. Another type of silent mutation is that the new nucleotide codes for a similar amino acids, because amino acids can be grouped together by their properties. There are some that are have more affinity to water. There are some that have more affinity to fats. There are some that are more acid. There are some that are more basic. And so if you have a substitution of at the amino acid level for one amino acid that really behaves like another one, it may just cause a silent mutation. And there also are parts of our DNA where are not genes, and a mutation in one of those sections, these non-coding sections, may not have an effect. Then we have a missense mutation, and this changes one or more amino acids in the protein, which then changes the protein shape. And proteins are very large molecules, and they do what they do because of their shape. So if the shape gets tweaked a little bit because an amino acid is not quite right, the protein might still be functional, but it no longer works perfectly, and that can cause problems. An example of this is sickle cell anemia. And so this is caused by only one wrong amino acid. If we look here, the wild type hemoglobin, this particular codon is in the DNA in the gene is CTT, leading to mRNA of GAA, which would code for glucine. Whereas in the sickle cell state, it becomes CAT, so that T has been replaced by an A, which results in the mRNA codon as GUA, which then puts valine into the hemoglobin of sickle cell, or people suffering from sickle cell. And so this wrong amino acid in the hemoglobin gives it a different shape, which then forces a change in the overall shape of the red blood cells, which is the reason we call it sickle cell anemia, because the cells that are carrying that hemoglobin tend to form this crescent shape or this sickle shape, which doesn't flow through blood vessels very well and can cause all kinds of problems. Even though it still can carry oxygen, it has other effects, and that's why this particular disease is an issue. And it's all due to just one single nucleotide being the wrong one. Then there are nonsense mutations. These cause a premature stop codon. If you remember from the transcription and translation activities, all proteins will have a start codon on the gene saying it's, this is where things start to build this protein, and then there's a stop codon in the gene that says this is where you stop building a protein. So if you have a premature stop, if it comes in too soon, then the protein ends up being too small because you've just lost a whole bunch of the amino acids that are supposed to be there, and these proteins are non-functional. So the sickle cell anemia hemoglobin is partially functional, but it has other issues because it changes the shape. But in this case, in a nonsense mutation, you just lose that protein as a functional part of the body's homeostasis, lose it entirely. Cystic fibrosis is caused by a nonsense mutation, which results in a malformed CFTR protein, which stands for transmembrane conductance regulator protein. And these CFTR proteins create channels in the cell membrane to allow chloride ions to pass through. Remember, our cell membrane, things have to go through it to get from outside the cell to inside the cell. And in cystic fibrosis, chloride ions are not able to move correctly because the channels are not present, because we've had a stop codon go on too soon. So our wild type examples on upper part of this diagram, and you can see that, the protein is supposed to be longer than what you get when you have a stop codon put in too soon. And then there are frame shift mutations. And these are when you have an insertion or deletion point mutation, which shifts the reading frame, or shifts those groups of 3 codons that are built from the 3 nucleotides, which completely changes the protein. So to use actual words, for an example, I see my picture is covered over some letters. Sorry about that. But let's assume that our protein is supposed to say something. We're using 3-letter words here because codons are in 3. And so we're supposed to say the fat cat ate the rat. So that would be the correct reading frame, looking at those codons in groups of 3. But if we have a nucleotide inserted, so if it's inserted right here, then instead of the fat cat, you get the fat, and then it loses completely, hakat tat ether. It doesn't make any sense at all. So the reading has shifted. These codons no longer make sense. The same thing happens with deletion. So here we have a deletion right in the very beginning. Our H gets removed, and so everything shifts over one space, and again, the words no longer make sense. These codons don't make sense because they're going to be putting, in this case, they're going to be putting in the wrong amino acids, but it's going to be completely wrong. It's not like one is wrong or the codon is a little bit, or the protein is a little bit too short. I mean, the whole thing is messed up. Frame shift mutations generally cause pretty severe disorders. Tay-Sachs disease is caused by a frame shift mutation, which results in one of these nonsense mutations, and you have an abnormal enzyme in the lysosomes of the cell that cannot digest the waste products in brain cells. And so basically fat will be building up in the brain cells, and it will become overflowing, and those cells will die. So individuals with Tay-Sachs rarely live past age 5 because it is such a severe disorder, not having that enzyme present at all, because it's a frame shift mutation and it just completely messes up the protein. Then the other type of mutations are chromosome mutations, and this is a change in the number or the structure of the entire chromosomes. So we have our karyotype here showing sort of a typical pattern of chromosomes, and so if you're missing a part of a chromosome, or you have an extra chromosome, or your entire chromosome is gone, that's going to affect the individual with those chromosomes. The first type of chromosome mutation is known as non-disjunction. And in non-disjunctions, the chromatids, those replicated copies of the same chromosome, do not separate during meiosis, which is cell division when you're creating the gametes. And so you end up with an extra copy of a chromosome in the cells. Probably the most familiar form of non-disjunction mutation is Down syndrome, where a person ends up with an extra copy of chromosome 21, so they would have 47 chromosomes instead of 46. Down syndrome has a particular facial appearance, and people suffering from it do have some level of intellectual disability and developmental delays, and might have other serious physical problems that need to have surgery, but it varies quite a bit from individual to individual. And I just gave you a picture here from an actor that is on the show called The Midwife, who performs regularly, who has Down syndrome. Then if part of the chromosome is missing, it's known as a chromosomal deletion. So an example of a chromosomal deletion mutation is what's known as Cre-du-Chat syndrome. This is caused by the loss of part of chromosome 5, so not the whole, there's still some of this chromosome present, but some part of it got broken off somehow in the process of meiosis when the sperm or the egg was being made. So people who have Cre-du-Chat syndrome, and it's called that because that's in French, that means the cry of the cat, and the sound of the babies make sounds very high-pitched like a kitten mewing. So these children, when they're born, their heads are smaller than normal, so you have microcephaly, and because of that, you have various developmental delays and may have malfunctions of several body systems. But again, it can vary quite a bit between individuals, between someone who may not live past the first year of life Though there are some, but there are some people with Cri du Chat who've lived up to their 50s. So the exact genes that are missing, the exact part of the chromosome that is missing, is going to then affect the lifespan and how this person is affected. And then there can be a chromosome completely missing. And the most common and the only one really that leads to long-term viability that I know about, that I've read about, is what's known as Turner syndrome. And in Turner syndrome, there's only one sex chromosome, and no other chromosome. So a person only has an X chromosome, be like an X0, not an XX and XY, but just an X, nothing else. So the persons typically appear and identify as female, but don't have fully functional ovaries, so they don't develop quite all of the secondary sex characteristics. There's other physical and developmental disabilities that typically are present, but again, things can vary quite a bit among individuals. And I remember a couple years ago reading about a student who was starting at USC who was 16 years old with Turner syndrome, and but obviously had no intellectual disability because they were ready to go to college at 16. So what is the significance of mutations? Well, there are a lot of mutations present. As I said, we're all walking around with mutations because most are neutral and just cause variation within populations. So the various eye colors, the various hair colors and hair textures, and I have a crooked little finger and the size of your toes, those are really just neutral mutations. They don't really make a big difference. But some mutations are harmful, and these are the ones that we tend to associate with genetic disorders because mutations form on your DNA, so then that's your genes, so it's a genetic problem. And there are, we are trying to develop therapies to deal with some of those, but some of these disorders really, because it's affecting every single cell in the body of an individual, they're not going to be able to be really successfully treated. You can't change every single cell in somebody's body. And some mutations are beneficial. There is an indication that some people have a mutation that allows them to be immune to the HIV virus, which would be a beneficial mutation since that is a pretty nasty virus. And we have plenty of examples in bacteria that have mutations that make them resistant to antibiotics, which is great for the bacteria, but not so great for us trying to treat that. And so mutations, have a variety of outcomes. The ones that we're most worried about and the ones that you tend to think about are harmful. So far as I know, there's no mutations that's going to turn you into a superhero. But hopefully this video helps you get a little bit of a better understanding about how your DNA and changes in your DNA can lead to a change in the protein, which is why we see a mutation in the individual.